Ever pondered over the process that goes into collecting specimens for scientific studies? It's a meticulous journey, and today we'll delve deep into this intriguing world. Picture this: a scientist, donned in their lab coat, is in the field or lab collecting samples. It's a process that requires precision, patience, and a deep knowledge of standard procedures. The first step is to determine what type of specimen needs to be collected. Is it a blood sample, a tissue biopsy, maybe a bacterial swab? This decision is crucial as it sets the stage for what comes next. Once the type of specimen is decided, the appropriate collection method must be chosen. This could range from a simple swab or needle prick for blood and skin samples to more complex processes like biopsies for tissue samples. The choice of collection method is influenced by the type of analysis to be carried out. For instance, if the aim is to study the DNA of a plant, a small leaf sample would suffice. The next step is the actual collection. This requires precision and care to avoid contamination. The scientist must wear protective gear such as gloves and face masks and use sterile equipment. The collected sample is then placed in a sterile container ready for transport. Transportation is a crucial part of specimen collection. Samples must be kept at the right temperature and humidity levels to preserve their integrity. Some samples, like blood and tissue, may need to be refrigerated, while others, like bacterial cultures, may need to be kept at room temperature. Once at the lab, the sample is prepared for analysis. This could involve staining for microscopic examination, culture growth for bacterial samples, or DNA extraction for genetic studies. Now let's summarize the key points about specimen collection. First, it's crucial to determine the type of specimen and the appropriate collection method. This is influenced by the type of analysis to be carried out. Next, the actual collection must be done with care and precision to avoid contamination. Protective gear and sterile equipment are a must. Transportation is a vital step that ensures the sample's integrity. The right temperature and humidity levels are critical. Finally, the sample is prepared for analysis in the lab. This could involve various processes depending on the type of sample and the intended study. So the next time you see a vial of blood or a petri dish in a lab, remember the meticulous journey it's been through. It's a world of precision, patience, and processes, all in the pursuit of scientific understanding. This, my friends, is the fascinating world of specimen collection.